In this video, we will talk about the phases of fertilization.
First, let's look at the penetration of the corona radiata. So, what is corona radiata? Corona radiata is a glycoprotein membrane that surrounds the oocyte plasma membrane. It is the innermost layer of cumulus oophorus surrounding the zona pellucida. Corona radiata act as a protective layer for the oocyte and also provide vital nutrients for the oocyte maturation. Next, we're going to look at how the sperm penetrate corona radiata. There are two aspects of the sperm that ease the penetration. The first one is hyaluronidase released from the acrosome of the sperm. Hyaluronidase, a corona-penetrating enzyme, breaks down mucopolysaccharides, the material that holds the cumulus oophorus cells together. The motion of flagella of the sperm also helps the penetration of corona radiata to be more effective.
Next is the penetrations of the zona pellucida. What is the zona pellucida? Zona pellucida is a glycoprotein layer that is surrounding the outer layer of plasma membrane of the oocyte, which causes the acrosomal reactions and maintains the sperm binding. Zona pellucida, it consists of 4 glycoproteins, which is zona pellucida 1, zona pellucida 2, zona pellucida 3, and zona pellucida 4. In human, zona pellucida 3 and 4 is responsible to block cross-species fertilization. For the acrosomal reaction to occur, spermatozoa must be recognized by a specific glycoprotein in the zona pellucida called zona pellucida 3. This is specific to each species. The acrosomal enzymes esterase, acrosin, and uraminidase are then released, digesting the zona pellucida and allow the sperm membrane to fuse into the oocyte membrane. After that, the permeability of the zona pellucida will change when oocyte and the sperm come in contact.
Next is prevention of polyspermy. What is polyspermy? Polyspermy refers to the fertilization of the egg by more than one sperm. This condition results in zygotes with greater than a diploid amount of DNA. This causes early embryonic defect and arrest of development. Two mechanisms that prevent the polyspermy are fast block and slow block to polyspermy. Fast block involves change in the electrical potential across the egg membrane, while slow block involves exocytosis of cortical granules in the process of cortical reaction. And the ion required in fast block is sodium ion, while slow block involves with calcium ion. The resting membrane potential is negative 70 millivolts. The inside of the cell is negatively charged with respect to the exterior. Once the first sperm binds to the egg, sodium ion rush into the egg and increase the membrane potential to 20 millivolts within 1 to 3 seconds due to the influx of sodium ion. The change of electrical potential across the egg membrane causes the egg depolarize. Then, sperm are no longer capable of binding with the eggs, preventing polyspermy. Slow block starts after 20 seconds of sperm attachment and is complete within a minute before end of fast block. The intracellular calcium initiates the cortical cranial reaction. Once the first sperm entry, the calcium level in oocyte rise and trigger the cortical reaction. The cortical granule fuse with cell membrane and release their contents into the perivitally space, or known as the cortical reaction. Among the contents of cortical granules are enzymes that act on the zona pellucida to prevent binding and penetration by spermatozoa.
Now, let's move on to the fusion of the oocyte and the sperm. The sperm's head and tail will enter the cytoplasm of the oocyte, and its plasma membrane will remain behind, meaning that the sperm's nucleus will get into the plasma membrane of the oocyte, and that's how sperm releases its genetic material into the egg cell. Also during the fusion, oocyte completed its second meiotic division, which will result in the formation of the mature ovum and the second polar body. After that, the formation of the pronuclei will slowly begin. What is pronuclei? Pronuclei is the haploid nucleus of either the sperm or egg before the fusion of the nuclei in fertilization. The chromosomal material of the sperm decondensates and enlarges. The sperm will also loses its flagellum, forming the male pronucleus. The genetic material will then reform to be 23 pairs of chromosomes. Next, the chromosomal material of the ovum decondensates and following the completion of the meiosis. Ovum will also extrude second polar body, which will become the female pronucleus, which also contains 23 pairs of chromosomes. As both the male and female pronuclei form, they unite to form a single group which represents prophase of the first cleavage of mitosis, and will then continue the rest stages of mitosis. Fertilization results in a lot of important things. Fertilization will stimulate the secondary oocyte to complete meiosis. As we all know, the oocyte is stuck in metaphase 2. This means that meiosis 2 will only be completed if fertilization occurs. Fertilization also restores the normal diploid number of the human chromosome, which is 46 chromosomes. It will also create variation in the human species as the maternal chromosomes and the paternal chromosomes will intermingle. The sex of the embryo can also be determined after fertilization by looking at the type of the sex chromosome carried by the successful sperm.
To conclude our video, we have learned about 3 phases in fertilization. Penetration of corona radiata, where the sperms uses hyaluronidase and tail movement to make the passage. Penetration of zona pellucida, helped by the enzymes esterase, acrosin, and neuraminidase, which involves an acrosomal reaction, and cortical reaction, where fast block and slow block are vital in prevention of polyspermy. We also learn about the fusion of oocyte and sperm cell membrane and formation of pronuclei. It is important for this knowledge to be shared so we have better understanding of our body.
That is all from us. Thank you.